Hello everyone, welcome to Critical. So today we are going to discuss about the chapter Photosynthesis in higher plants. So in this chapter there are totally 10 topics. 10 topics. We are going to see how photosynthesis are occurring in plants. Okay. So here what do we know generally about photosynthesis? Here is the first topic.
From our last standard what we studied that we are going to see.
Then early experiments which made on
19th and the 10th. Okay.
Where does photosynthesis take place?
Yeah, leaves that we know. Okay. We are going to read it.
Yeah.
Okay.
How many pigments are involved in photosynthesis? Okay, that's what we are going to study. And the fifth topic is what is light reaction?
Okay.
And
the electron transport.
It is the sixth topic, and in electron transport there are three subtopics: splitting of water, cyclic and non-cyclic photophosphorylation, chemiosmotic hyposynthesis.
Okay.
Hypothesis. Okay. So here, where are the ATP and NADPH used? It is our seventh topic. In seventh topic, there are two subtopics. The first one is the primary acceptor of CO2 and the Calvin cycle. And the eighth topic is the C4 pathway. Ninth one is photorespiration. 10th one is factors affecting photosynthesis. In that there are four subtopics: light, carbon dioxide.
Yeah, we already know, but we are going in detail.
Yeah.
How many temperature required, water,
Temperature
light,
and water.
carbon dioxide.
So these are the 10 topics we are going to discuss. So we will go with the first one. What do we know generally about photosynthesis? Okay. So now we are going to see first topic, which is what do we know generally about photosynthesis and what experiments we have done in our previous classes. Okay. So we are generally seeing about these experiments. So photosynthesis is generally a physicochemical process.
Physicochemical process. Okay.
It is both physical and it is chemical also. Okay. So here in photosynthesis there is three main factors are required: chlorophyll, light and carbon dioxide.
Chlorophyll, light and carbon dioxide. Okay.
So with these three only, these three are very required, are very important for photosynthesis to occur. Okay. So here we are going to see about the first experiment. Okay, this is the experiment for starch formation.
Okay.
Starch is nothing but the sugar. Okay, sugar formation in these plants, especially in the leaves. Okay. So two leaves are taken. Okay, two leaves are taken generally. One is variegated leaf. Variegated leaf is nothing but it is covered leaf. Okay, and one exposed leaf. Exposed leaf is that exposed to light conditions. So here photosynthesis occurred only in the green parts of the plant.
Green parts. Okay.
Green parts of the leaf in the presence of light. So here what we are coming to conclude is both chlorophyll and light are exclusive for photosynthesis by this experiment. Okay. So while coming to the second experiment, which is half leaf experiment. So here a part of leaf is covered. Okay, a part of leaf is covered with KOH, potassium hydroxide, or soaked with cotton. Okay, so this potassium, the cotton is soaked in potassium hydroxide, and it is covered, and the half part of the leaf is covered by this cotton.
Okay.
So what does this potassium hydroxide do? They absorb all the carbon dioxide. Does not leave any carbon dioxide entering the leaf. So the other half is exposed to air. Air means which is having both carbon dioxide, oxygen, nitrogen and everything. So here the exposed part gave positive for starch.
Okay.
Okay. So the leaf which was soaked with cottony KOH, potassium hydroxide, it does not show positive for starch. So with this what we can conclude is carbon dioxide is necessary to perform photosynthesis and to prepare starch. Okay. So therefore carbon dioxide is required for photosynthesis. Okay.
Carbon dioxide plays a main role.
Yeah. So we came to a conclusion that carbon dioxide, light and the green parts, chlorophyll, are important in the process of photosynthesis. So next we go to the next topic, which is the early experiments made on photosynthesis. Okay.
Early experiments.
So now we are going to see about early experiments, okay, which were done in the
Early periods.
yeah. So here the first experiment was done by Joseph Priestley. Okay. So he only discovered oxygen in 1974.
He only discovered oxygen. Okay.
Joseph Priestley discovered oxygen in 1774, and he observed that when a candle was glowing in a closed space, closed area, after some time the candle was off.
Because it needs oxygen to burn.
It extinguished. And he also kept mice. Okay, mice in closed space. So after some time this mice also died. Okay. And when he kept a plant, mint plant, inside that closed space, one mint plant, one candle, and this mice. So the candle and mice were alive. Mice were alive, and the candle was glowing there. Okay. So he only observed that. So what conclusion did Joseph Priestley did here?
He kept the three atoms
So
together.
he just noted that the plant could purify the air, which the burning candle, and the mice were, it was distracting the air. So it purified the air. He thought like that. Okay. So next, the second scientist, who is Jan Ingenhousz. Okay. He performed some experiments. So he placed a plant once in dark and once in sunlight. Okay. So he found that sunlight is essential for plant processes.
Okay.
Like he took aquatic plants. So these aquatic plants under bright sunlight, near the leaves of these plants there were bubbles coming out. Okay. So why the bubbles are coming out?
Because of the oxygen.
Because of the release of oxygen. Okay. So he came to the conclusion that green parts of the plant releases oxygen.
Okay.
Okay. So now third scientist, Julius von Sachs. Okay. So what he did, he experimented that. By his experiments, he concluded that production of glucose takes place in the leaves, and it is stored as starch. Okay, in the leaf, in the green parts of leaf. Okay. In a special structures called chloroplasts. Chloroplasts inside that thylakoids. He proved that one. Okay. So next we will see some other scientists who did some more experiments on photosynthesis. Okay. Now in early experiments, we saw about 3 scientists who performed who performed some experiments on photosynthesis. So now we are going to see the next scientist, who is T.W. Engelmann. So he used a prism. Okay. He used a prism and split light into its components, which is VGR. So here he split light into its spectral components. He kept a green algae in that light, in that split light. Okay, he kept a green algae, Cladophora. Okay, it was a green algae which contains pigment in them, chlorophyll and everything. So placed in a suspension of aerobic bacteria. Okay, it was placed in a suspension of aerobic bacteria. Okay, so here this bacteria, Cladophora, it is used to detect the site of oxygen evolution. Okay. So since it is aerobic, it needs oxygen to live. So it finds a place where oxygen is liberating. Okay, in that splitted light prism. Okay. So here, so the bacteria accumulated in blue and red light of the split spectrum. Okay. So this bacteria Cladophora accumulated in blue and red light in VGR. Okay. So why it is accumulating in blue and red light?
Because the oxygen is available in the blue and red light.
No, it is because the wavelength of the green light. Okay. So in Lucia, all the colors are dependent on wavelength.
Yeah.
So this green light have a particular wavelength which is suitable for blue and red light in the splitted spectrum. Okay. So here this absorption spectra of after coming to know that blue and red light of the split spectrum, then absorption spectrum of chlorophyll A and B was discovered. Okay, it is the absorption spectrum of chlorophyll A and B. So now after these experiments in the middle of 19th century, plants could use light energy to make carbohydrates from CO2, carbon dioxide and water. It was proved. Okay. So from these experiments, it was proved that by getting the light energy, carbon dioxide and water, plants could make carbohydrates, which is the sugars which are starch, which are stored in the leaves. Okay. So here after this statement, Cornelius van Niel, he was another scientist. So he, what does he prove is oxygen is liberated from water only, not from carbon dioxide. Okay. So plants absorb carbon dioxide and even water, but the oxygen liberated is coming from water and not from carbon dioxide. He discovered this. Okay. So here by several radioisotopic techniques, later it was proved that this oxygen is coming from water only and not from carbon dioxide. Okay. So for this he used sulfur bacteria, green sulfur bacteria. Okay. So in these sulfur bacteria, in order of this 12 H2O, it contains 12 H2S sulfur here. H2 instead of O, it has S sulfur. Okay. So after the reaction occurs with the light, so this sulfur was seen with 6S2, not 6O2. So therefore it was identified this H2O water molecule is responsible for releasing oxygen. So therefore sulfur was separated. 6 sulfur molecules were coming out. Okay. With this experiment, Cornelius van Niel found that oxygen is liberated from water only and not from carbon dioxide. Okay. So we will go to the third topic, which is where does photosynthesis take place. We are going to know where does photosynthesis takes place. Okay.